I would like to talk about the olfactory abilities of birds and their interactions with plants. And as it was said, I will be combining the research from temperate regions and from the tropical regions. And as you all know, the birds need to eat something, and many of them are insectivorous, and they are searching for the insect in the dense vegetation. And for that, they really need to have good orientation skills and good ways how to search for the insect. And actually, the insect tries to, of course, escape because they don't want to be eaten by the birds. But unfortunately, there are these nasty plants who are giving them away. So there are three trophic interactions between plants, insect, and the birds, which I am studying and which I will be introducing in this talk. The interactions between the plants and insects started to develop 500 million years ago, when the first insectivorous insects started to eat the plants, and plants started to defend themselves. And they developed various ways how to protect the green foliage by mechanical defenses, by chemical defenses, or by simply being thick and disgusting for the insect. But for me, the most interesting are the interactions which are tritrophic, where the other trophic level is helping the plants to remain green, not to be eaten. And when we talk about the plant defenses, we can call them constitutive. Those are the defenses which plants have always present in themselves. And these things are actually pretty visible because the plants invest so much carbon into the protection of their green foliage that, for example, you can see the chemical compounds being released to the air in, for example, Smoky Mountains or in Blue Mountains in Australia, where I used to work. So these are the chemical compounds which the plants on the everyday basis release to the air, and they signal something. But the most trickier things and the interesting for us are the induced plant defenses, which the plants start to induce only after they are eaten by insect. These chemical signals serve pretty well to other trophic levels that the insect is there, and these chemical compounds are very specific and interesting for the trophic interactions. And as it's indicated here, so the plant is induced somehow by insect which is feeding on the plant. It's indirect interaction which leads to the parasitoids or to some other predators helping the plant to get rid of the insect. And all these trophic cascades within the plant are managed by various acidic compounds, for example, jasmonic acid or salicylic acid. The jasmonic acid is typically released in the plant when the chewing insect feeds on the plant, and salicylic acid when the sub-sucking insect is sucking on the plant. And they are causing different things happening in the plant, so the birds or other predators can distinguish what they can be searching for. And all these chemical interactions in the plants are very specific because the insect is having some compounds in their saliva. These are being released into the cells which are being mechanically broken during the chewing. And the plant is always releasing a very specific smell based on what insect feeds on it, or also on how many insects feed on them. But then you can imagine that the things are very complicated because, for example, younger and older caterpillars, they have got different saliva. So the plant induces different chemical compounds based on how old caterpillar feeds on it. And for example, if two different species feed at the same time, the chemical compounds are being mixed and the signal is very specific. And it has been known from 1983 that the plants have such chemical compounds and defenses. And in 1990, it was shown experimentally that the parasitoids are attracted to these chemical compounds. This is because they need to find their specific hosts in which the caterpillars, which the eggs can easily develop. If they don't find the correct caterpillar, the eggs within will die. So parasitoids really need to find the specific chemical compounds based on the plant, based on the caterpillar feeding. But the question whether the other predators could be using these chemicals for orientation was here. And it was not until 2008, 2009 when the first experiments appeared. And this is when actually I was finishing my master's degree, where we started with these experiments. And I will be showing now some of them, but I know that all of you believe or know that the birds orient themselves also very much also visually. They're colorful, they signal colors, and they recognize them pretty well, so they for sure use the visual orientation very effectively. On the other hand, their olfactory bulbs are pretty small, very tiny in contrast what they had before and how they were getting smaller and smaller during evolution, which made many people believe that birds can't smell generally anything. And they are really tiny these days, just tiny bit of the brain left there. So still the question is whether it's useful for something or not, and specifically how much the birds use the vision, acoustics, and olfaction all together for getting the proper signal. There are birds which have still pretty big olfactory bulbs. For example, snow petrels, yes, kiwis, turkey vultures, and the songbirds have very tiny olfactory bulbs. When we look at the snow petrel, this is the bird which is feeding on very basic food on the krill in the sea. And they recognize the places rich in krill based on the sulfidic compounds which are released from the water after the krill feeds on the algae. So very typical, very similar interactions, which I will be talking about, but just happening under the water, and very simple chemical compound. Then we have the vultures, who are feeding on dead, decaying meat. Again, very simple chemical compounds released in the air, which they find effectively. And then we have, for example, the homing pigeons, still birds with quite large olfactory bulbs. And the birds, the pigeons can navigate themselves to their home range based on those constitutive plant defenses or constitutive plant smells. For example, they can recognize that here is the smell of pine forest, smell of the water, and smell of the feeds. And based on these smells coming from directions, they can navigate themselves home. But then, as I was showing on the graph, the songbirds are having even much smaller olfactory bulbs. So do they use them for something? And so we were doing many experiments with songbirds, especially gray tits, blue tits, and we were observing through the behavior experiments how they perceive the chemical compounds. Most of the experiments was conducted in kind of Y-shaped aviaries, where we were providing them different signals, either smell or visual signal, through the different doors birds were able to see or not see, or just smell the signal coming from the plants. And in the first experiments, we were using the live insect, which was very tricky. So what we did is that we took the insect, placed them on the trees, started the chemical reaction in the plants started to produce them, and we were offering them to birds, seeing whether they like them or not. And in our very first experiment, we were offering, together with my student Anna Mrazova, the signals like control versus the chemical and visual signal together. According to the expectation, both bird species pretty much preferred the both signals, blue-tit significantly more than the gray-tit, but still there was some slight difference. But when we offered them the visual signal versus chemical signal separately, they behaved against to our expectation, and they all went for the visual signal, which is, of course, they can see the holes, why they would not go there, and they were totally forgetting about the chemical signal. So I said, okay, they don't have chemical orientation, forget about this. But at the very same year, our colleague Luisa Amo in Spain conducted very similar experiment with the gray-tits close to Madrid, and what she showed is that their bird preferred the chemical signal over the visual ones, which was like, okay, this is very weird, our birds do opposite things than your birds. We went together, conducted the experiments with her birds, and indeed we confirmed that at least the Spanish birds react well when we do the experiment well. So over the time, we learned that the system is very tricky. We really need to match the proper age of the caterpillars, proper age of the leaves, and only after that, the birds, when they are in a good mood, they behave well, and they can react to the chemical signals pretty effectively. But most of the time, when you just catch them from the wild, put them to the cage and want something from them, they don't do anything. They just try to escape or die, and they are not very cooperative. So to ease the further experiments, we avoided the direct experiments with the insect, and we jumped directly to the metal jasmonate. Because if you take the plant hormones and you spray them on the plants start to make the exactly same things as the insect would be eating them. So we removed some of the factors, and because the birds were very much stressed in the cages, we started to do the experiment in the wild. And we were working on English oaks and the winter oaks, and we were spraying them with the metal jasmonate and using them as the control. And after that, we were observing through the method with plasticine caterpillars whether the birds are attracted to the birds or not. So these caterpillars are like fake caterpillars made out of plasticine. When the bird is attracted to them somehow, they are leaving the bite marks on them. So we can easily compare if the tree sprayed with the hormone is attractive or more or less attractive to the birds through this method. It worked pretty well according to our expectation. Whatever plant species of oak we sprayed, birds preferred the one which was sprayed with the metal jasmonate, and we also chemically compared that these trees expelled to the air much stronger chemical signal. And this was an experiment with rather small trees planted in quite open space. But we still don't know what is happening in the bigger trees where there are massive amounts of the foliage, various insects eating at the same time. What is actually happening, to what distance the chemical signal can work, whether it spreads through the branches to the whole tree, how this could work. So still lots of space and topics which can be studied here. And as I used to work before in the tropical forest the most, I was wondering how this can work in tropical forest. We had very simple experiment with tiny oaks in Czech Republic, and tropical forest is massive amount of foliage, lots of chemical compounds, smells in the air. So how this could work? Moreover, there are rather low densities of arthropods per meter square of the foliage. So the insect, there is lots of it, but per meter square of the foliage, there is only a little bit of it. The understories are dark, dense, birds have difficulties to orient themselves. And I think most importantly, the leaf in the tropical forest is there forever. We have a leaf which is on the tree for 5 years. But after 2 months, it's not chemically active anymore. So it's tough, disgusting, no one eats that, it's just surviving there. So it's very difficult to study somehow the chemical communication in this system. And this is just to show that really the abundances of the arthropods are pretty low based on our data, but there is indeed lots of foliage in the tropical study sites. So we have quite a different system, and to study it, we first went to the elevation gradient of Mount Wilhelm in Papua New Guinea. And this is the study site where I worked a lot before, where I studied bird communities, so I know a lot about them. And it's a wonderful study site or gradient which spans from 200 meters above sea level to 3,700 meters. Very different habitats, dense understories, open habitats, tree line, very cold conditions at the tree line, very hot conditions in the lowlands. So wonderful experiment where I can see what's the impact of the birds and interactions with the plants. And because I said that the chemical released by plant species are very specific, we were of course searching for a plant species which is widely distributed. It's not so easy. There is not many plants which would be growing all the way from the lowland to highland, and we were very lucky, and I mean very lucky. We found a Ficus haliana which grows up to 3,000 meters above sea level. It was wonderful. We were running up and down. There was sometimes swimming involved, lots of swimming even in the cars. There were no roads, or there were roads sometimes. So lots of fun to find that plant species. And we used again the similar methods with the plasticine caterpillars to survey the attractivity of the trees to plants. And for the first experiment, we went for the visual orientation of the birds. We just made holes to the leaves. And we were wondering whether these holes in the leaves will be attracting more birds to the trees or not. Here are the results. Not too much, actually. There is little increase here, only 2 study sites, and only at 2 study sites the holes in leaves attracted more birds than the controls. So we said, okay, maybe it will work, but let's try the chemical way later on. And there we worked at 4 elevational study sites at 200, 700, 1700, and 2700 meters above sea level. We again sprayed the Ficus haliana with a methyl jasmonate, and we attracted significantly more birds in lowlands. Also in highlands, but not as many as in the lowlands. So this was pretty cool, it worked. But as I said, we had real good luck, because after that we tried several more plant species to induce, and nothing worked. We managed with Ficus haliana pretty effectively, with syzygium and other ficus species, no luck at all. So we still don't know how much the chemicals actually work in the tropical forest. And after these experiments, I actually, if I can be personal, got pregnant, got stuck in the Czech Republic. It was the end of my PhD. So I started to focus on similar topics, but with the conditions which I was given at the time. So I was combining the orientation of the temperate birds towards the tropical and temperate birds, and I was investigating whether the birds have this ability to smell chemical signals, innate or not. I was working with the great tits, and then I picked the tropical Psidium plant and Ulmus temperate plant, which is quite uncommon here, so the birds did not have strong evolutionary contact with it. And together with my assistants and student assistant, which was helping in that lab at the time, we were again planning experiment in which we planned to show the birds control tree without a chemical signal and the chemically induced tree, which produces the chemical signal. But at the time, there would be, of course, no caterpillars, no holes visible. We just went for the chemical signal. And now here. So we took 30 gratids and we split them into 3 groups. 10 of them, we were teaching how to associate the food with the smell of elm. 10 of them, we were teaching how to associate the smell with the food with the smell of guava, the psidium. And 10 of them we kept untrained. Untrained means that they were living in the aviaries where there were only dead branches and they didn't come into any contact with the green foliage. Those trained ones, they were always getting the chemically induced plants, and there were worms, caterpillars pinned on them, so they got the reward and they associated that smell, specific smell, with the food. And our hypothesis was that if the ability to recognize chemical compounds is innate, of course, no matter what we teach them, all of them will prefer the chemical compounds. If they are able of generalization, the not taught birds will be acting weirdly, but any trained bird will be able to react to the chemically induced plants positively. And then the third hypothesis is that these signals may be very specific, so only the elm-trained birds will prefer elms, and only guava-trained birds will prefer the guavas. And of course, non-trained, again, nothing. You can guess the results, but after many, many months of training, because we realized the birds are pretty dumb altogether, they didn't want to learn anything. So after several months of training, those non-trained birds behaved exactly as we expected. They were flying from tree to tree, side to side, 50% here, 50% there. They didn't know what to do because they were not trained to foliage at all. When we put the elm-trained trees to the experiment, they typically preferred the induced elm, the light blue. Right? Whatever combination, whenever there was induced elm, they kind of went for it, except there was the guava. And when we tested the guava-trained birds, again, they preferred very much induced guava. So we found that the birds really need to learn, need to have the previous experience with the chemical compounds, positive associations, and after that, it's pretty much specific, but they still prefer the guava a little bit more than the elm altogether in ill experiments. Maybe because the guava had a very strong signal from beta-ocimene, which is typical compound in many plants, woody plants. So there might be some generalization, but very likely it's a specific response to the plant compounds. So this was the experiment which I played with a bit in Czech Republic. And after that, Chido was a bit bigger, and I was allowed to go back to the experimental sites, to Papua New Guinea. And at the time, I started to remove the birds from the study system and see what happens if we get rid of the birds altogether, just in case they die out or something like that. So I returned to Papua New Guinea, and along the same elevational gradient of Mount Wilhelm, together with my assistant, we started to do many assistants, sorry, not only one, many. We started to exclude the predators. Sorry, I just weird button. We kept some trees, of course, as a controls. We had some trees from which we excluded ants, because ants are pretty good predators, people say. We excluded them by the glue applied to the trunks. Then we excluded vertebrates together by cages which were permanently closed and standing around the trees. And then we combined the treatment, glued it all together, excluded to the cage, nothing could get in. And then the most funny part, we were excluding birds and bats separately. It's just being done by the cage, which you pull up and down every day in the morning, in the afternoon, depending on what you want to get rid of. This is what we were doing at many trees along the elevational gradient. This is what I actually drew to my grant proposal when I was still very pregnant, and this is what my assistant started to build in the tropical forest. So it was cool. And this is how the cages look like. This is actually in Leipzig. You will see later on. So the cages were 3 meters high, 2 by 2 meter big. We used hundreds of them. And in Papua New Guinea, we were building them from the PVC pipes at the site. Sorry, the color somehow changed, but we used more than 8 kilometers of the water piping and 6,500 meters square of the fishing net to conduct the experiment. And it was a great fun. And we were working with like 600 trees altogether. At the beginning, we just marked them, put the exclosures, and at the end, we were slowly lowering the saplings to the mosquito net, spraying the insect with the mortane insecticide, killing it or slowing it down, catching that, and counting how much insect was there at the end of the experiment after 6 months. And this is how the assistants are collecting the insect. Insect all fine, but I already said that in the tropical countries, there is the leaves on the trees all the time. So at the beginning, when we start the experiment, there is already some herbivory damage on them. So to calculate what is the effect of disappearance of birds on plants, we had to number the leaves, scan them at the beginning or photograph them, and 6 months later we scanned them and calculating how much more herbivory appeared on the leaves during our experiment. This is not fun. And altogether, along the elevational gradient, just to be a little bit faster, and along the elevational gradient in Papua New Guinea, where we excluded the ants, nothing happened generally. This is the change in arthropods. So when we excluded the ants during the size season, nothing happened. When we excluded them during the wet season, slightly the densities of the arthropods increased. But when we excluded all predators together and vertebrates only, the abundances of the arthropods increased significantly. So this is 6 months experiment, and abundances increased by 60–70%. Even higher change was happening in the herbivory damage when there were no vertebrates around the trees. The herbivory damage increased up to 500 times, but in average around by 100% more, so nearly doubled during the experiment. So for us, it was pretty good. We showed that without the birds, the trees would be very often quite eaten. We of course don't know how the balance would be establishing, for example, with mesopredators, which started to increase their abundances when the birds were gone. So there are many more interesting interactions with spiders and birds happening within this experiment, which is now difficult to show. But when we look to the birds and bats experiment separately, birds had typically higher effect than the bats themselves, but birds and bats didn't protect the plants too much in the lowlands, and they protected them very much in the higher elevations. This is quite interesting because a few years ago, before that, during my PhD, I showed that the highest abundances of insectivorous birds are really in the higher elevations, and this is pretty much matching what effect the insectivorous birds have on the plants, despite these experiments were nearly 6 years apart. So I think that there is, that matches pretty well. And after these experiments, We were actually extending them to the latitudinal gradient because we enjoyed very much building the cages and counting leaves. And I proposed, proposed to work originally in Tomakomai, in Hokkaido, in China, in Malaysia, Papua New Guinea, in drought tropical forest in Australia, and in quite dry eucalyptus forest in Sydney. But when we were supposed to go to Malaysia, unfortunately COVID happened and we could not get out of Europe. So we exchanged that study site to Leipzig. Why these study sites? It's all complicated because as we conducted all these experiments before only in the forest understories, which are pretty terrible sites, we this time wanted to go up to the canopies. So we wanted to get, with the help of the canopy cranes, up to the trees, to the crowns, see whether different things are happening there. And this is why we were dependent on the study sites which have a canopy crane present at the site. So these study sites altogether, and very nice results these are the study sites from Leipzig, the northernmost, all the way to the southernmost study site. Wherever we went, the exclusion of the bats, the yellow line, had a little bit lower effect than the exclusion of the birds itself. And these exclosures we were conducting only for 1 month, 2 times for 1 month, because it was a bit complicated to keep the people working for a long time, for every day in the morning and in the afternoon. So even after such a short experimental time, 1 month, we are able to detect quite large changes in how important these predators, insectivorous predators, are for plants. But of course, this is at the background much more complex, because as soon as we get rid of the predators, the plants start to protect themselves through calling for the help, and they are exhausting themselves by producing these chemical compounds to the air, despite no one can come and help them. So altogether across the globe, when we summarize all our experiments, we can see in the blue numbers that when we would lose the insectivorous birds, we would end up with much higher densities of the arthropods on our foliage, and all the trees would suffer much higher herbivory damage over the time. And the highest damage was here in southern Australia, was by 500% more herbivory over the 1 month long experiment, which is to me absolutely crazy. And in conclusion, I hope that I showed that that we have insectivorous birds which are very likely using the olfactory during searching for prey. Maybe not all of them know it, but we have got experiments just from few plants, from bird species, and we need to do more because it's very difficult to do experiments with other bird species. And it's very likely that these olfactory interactions between birds and plants are less significant in the tropical forest than in the temperate forest, because in temperate forest the leaves are much more active during the spring, there is much more insect, that's why the interactions might be stronger. There are the bird species which might be relying on very simple chemical compounds, and these might have these abilities innate, while those generalists seeking different complex compounds might need to learn that through the positive associations with the food. And I hope I showed that the birds and plants have quite tight relationships. They are helping to each other. They were developing over the hundreds of million years, these associations, and when we would lose the birds, the plants would be suffering, and the other way around. And these damages which the plants are suffering when we lose the bird just for several months completely are very significant. And by this, I would like to thank to all my collaborators who were working on the study sites, and of course to you for your attention. And I will be very happy to take any questions, because it was rather quick and rough, so there is many details which need to be answered. Thank you.